Hi, today we're going to talk about blood vessels. Blood vessels are the tubes that carry blood away from the heart towards tissues of the body and then returns the blood via the veins. So let's look at some basic definitions. So arteries always carry blood away from the heart. They empty into smaller vessels called arterioles and then eventually into capillaries. We'll learn today that capillaries is where all the gas exchange occurs, and then these capillaries merge to form venules, and then eventually veins. So a definition of vein is carrying blood to the heart. So this is a circular pattern that exists. The circular pattern of flow to and from the heart constitutes the blood vessels, which are part of the cardiovascular system. So let's break these down in a little more in order. So here's the aorta, and then these aorta branch into smaller vessels called arteries, which branch into even smaller vessels called arterioles, and these merge to form capillaries. So we'll learn that capillaries is where all the gas exchanges occurs between blood and surrounding tissue from the walls that are very, very permeable. Then eventually these are going to merge to form venules and veins, and then into the superior and inferior vena cava. So other terms we have to learn is the term vasovasorum. So vasovasorum exists on large blood vessels which have their own blood vessel circulatory pattern because they're so thick. The term angiogenesis means the growth of new blood vessels. So let's look at the basic structure of blood vessels. So blood flows through the lumen. These are these open spaces inside the lumen. You can see a difference between an artery and a vein, and we'll discuss these in greater detail. So the walls of the blood vessel consist of three major layers. The first one is called the tunica interna, which is made up of endothelium, and it has a basement membrane. The second layer is the tunica media, which consists of smooth muscle, which is controlled by the autonomic nervous system. And the most external layer is called the tunica externa, or the adventitia, which is formed by elastic and collagen fibers. So we'll discuss the differences between an artery and a vein. We'll look at them in cross-section, where you could see immediately the artery has a very small lumen, which is where the blood flows, and the vein has a very large lumen. And then you compare it to the lumen of a capillary, which only has a basement membrane and an endothelial layer. So let's break these down in a little more detail. So the tunica intima forms that inner lining of the blood vessels, and this is what has direct blood flow contact with the blood flow itself. You can see it right here, how it's larger in the vein and smaller in the artery. So within this, it has this endothelium layer, which is the layer that's continuous with the endocardial lining of the heart. Within the tunica intima, we could see this basement membrane, and the purpose of this is it's going to provide support, a physical support for that inner layer. In this layer, we also have a basal lamina, which anchors the endothelium to the underlying connective tissue. It also guides the cell movement during tissue repair of the blood vessel walls. The arteries have this internal elastic lamina, and you could see that the veins do not. So this is a boundary between the tunica interna and the tunica media. And you can see it has kind of a Swiss cheese appearance, which facilitates the diffusion of molecules throughout that region. The next layer in is called the tunica media. So in arteries, it's very thick, as you can see it here. So the role here in veins, it's thinner. You can see it right here. So the primary role of the smooth muscle is to regulate the diameter of the lumen, which is this inner layer. So smooth muscle contraction is regulated under the autonomic nervous system, means it occurs automatically. So the sympathetic nervous system, which is the fight-flight version of the autonomic nervous system, causes vasoconstriction. So what does vasoconstriction means is decreases the diameter of the lumen, versus vasodilation is increasing the diameter of the lumen when the sympathetic stimulation has decreased. The external layer is called the tunica externa. So this is the outermost covering, and this is primarily made up of elastic and collagen fibers. We talked about the vasovasorum in the past, and these are where we have vessels on vessels, specifically on large vessels like the aorta. So let's look at the comparison between an artery and a vein. So first of all, the lumen of the artery is going to be smaller versus the lumen of the vein. Secondly, you can see that the artery is going to have an internal elastic lamina and an external elastic lamina. You can see the artery has a very thick tunica media, where the vein has a really small one, and actually the thickest wall on the vein is going to be the tunica externa. You could compare this to a capillary that only has that basement membrane and the endothelium. Therefore, molecules via diffusion can pass through easily. So terms vasodilation, dilation means opening it up, which is the opposite of vasoconstriction. Here, you can see. So vasoconstriction, the vessel diameter decreases, stimulated by the sympathetic nervous system. The arteries, we're going to break down each of these vessels and discuss some of the details of each of them. So arteries, again, the definition is carrying blood away from the heart, not always oxygenated. In this situation, the walls are going to stretch to accommodate a high blood pressure that exists in arteries. So they have high compliance. What does that mean? That the walls can easily stretch out without tearing. So with the sympathetic stimulation, we're going to cause vasoconstriction of vessels. So in contrast, when this sympathetic stimulation decreases, it'll cause vasodilation. So the elastic arteries, when we're talking about arteries, there are two main sets of arteries. The elastic arteries are large. They're the largest of the arteries. They have the largest diameter when we're talking about all the arteries, and they have very well-defined internal and external elastic lamina. So the elastic lamina provides their ability to stretch out and come back to its original shape. Some examples of these elastic arteries are going to be the aorta, the pulmonary trunk, the branches off the aorta, which includes the brachiocephalic, subclavian, common carotid, and common iliac arteries. So what happens with this elasticity of these fibers, as they stretch out, they're going to store mechanical energy. Therefore, they function as pressure reserves. So remember that. The elasticity of these fibers will store that mechanical energy and then contract, propelling the blood onward. So that's why we call them pressure reserves. Muscular arteries are the medium-sized arteries. They're called distributing arteries. So these arteries are going into the muscle tissues, and they're capable of greater vasoconstriction and vasodilation to rate the rate of blood flow through targeted tissue. So the term vascular tone means that it stiffens the vessel walls. So the ability of muscles to contract and maintain the state of partial contraction is called vascular tone. The term anastomosis. What the term anastomosis means, it's the union of branches of two or more arteries supplying the same body region is called an anastomosis. So arteries that do not have an anastomosis are called end arteries. When we have an obstruction of an end artery, this can result in necrosis or death of that tissue because there isn't an anastomosis or another branch supplying that same region. So then we move on to the next set, which are called arterioles. So arterioles are the smallest arteries. They're actually microscopic, and they're important because they're regulating the flow of blood into the capillary networks. So their function is regulating blood flow from the arteries into the capillaries, and they also in turn regulate blood pressure. So let's discuss some terms. The met arteriole, this means the terminal end of the arteriole, and at that region is the met arteriole capillary junction. Precapillary sphincters controls the blood flow into the capillary, so that's important for controlling. So then understanding these concepts, we call arterioles as resistance vessels. So they play a key role in regulating the blood flow from arteries into capillaries. So here's an image. Here is the arteriole, and then the met arteriole. This is the end of the arteries before it connects to the capillary bed. And these precapillary sphincters monitor the blood flow into the capillaries. So we've reached the capillaries. So capillaries are considered exchange vesicles. So the capillaries are microscopic, and the blood flow through these capillaries is called a microcirculation. So they're exchange vessels. So their primary function of capillaries is to permit the exchange of nutrients and waste between the blood and the surrounding tissues. So the capillaries themselves are made up of an endothelial tissue and also a basement membrane. So this endothelial tissue, as you can see, it looks like simple squamous epithelium. So it's perfect for the exchange of molecules. So the blood flow from the met arteriole through the capillaries is through the into the postcapillary venule. So the flow of blood from the met arteriole through the capillaries into the precapillary venule. So the primary function of these capillaries is to exchange substances. Capillaries exist almost near every cell in the body, but depending on where we're at will indicate if we have more or less of them. So again, let's review this. Here is the arteriole, and then this is the end, the met arteriole. You can see these little smooth muscle sphincters that control the flow of blood into the capillary. Here's the capillary bed. And then going beyond that, we have the postcapillary venule, and then into the muscular venule. So sometimes we have what thoroughfare channel. This is where we close everything off and we direct the blood directly from the artery to the vein. So at the capillary, this is where all the exchange occurs. So often what's here is the capillary. You can see the red blood cells. So what is being expelled? Often water into the interstitial space, amino acids, oxygen, and nutrients as of glucose. What's being reabsorbed, which would include plasma proteins, water, waste products, and CO2. And here's an image of the arteriole. The precapillary sphincter closes off, therefore allows the blood to go directly through a thoroughfare channel from the artery, the arteriole, into the venule. So there's three types of capillaries. The most common one is the continuous capillary. So the plasma membrane of the endothelial cells of this form a continuous tube that is interrupted by intracellular clefts. The second one, which is the fenestrated capillaries, have little pores between neighboring plasma membranes. We see this type in kidneys, small intestines, the ventricles of the brain. And the third one is very porous. This is called the sinusoidal. They have really wide and more tortuous capillaries whose walls contain large pores and intracellular clefts. We see these often in organs such as the liver, the spleen. So on our way back, the first vessel away from the capillaries are the venules. So they have thin walls. They often do not readily maintain their shape. So they drain the capillary blood and they're returning the blood to the heart. So venules initially receive blood from the capillaries are called, the venules that receive the blood from the capillaries are called post-capillary venules. So these eventually start to get larger as it gets away towards the heart and eventually go into veins. So as we look at the veins compared to the arteries in this image, the veins have 3 layers as the arteries. The tunica intima is thin compared to the artery. The tunica media is very thin compared to the artery. It does have smooth muscle in this area. And of the 3, the tunica externa is probably the thickest. It has collagen and elastic fibers. Veins do not have that internal and elastic lamina. They're not designed to produce a lot, to withstand a lot of pressure. So the one thing that you will notice when we're comparing them, that the lumen of the artery is very small compared to the lumen of the vein, and you can see how the walls almost kind of collapse in versus the tunica media in the artery. It makes it very substantial. So how does the venous blood get back to the heart? Often it's controlled by the pumping action of the heart and also the skeletal muscles as they contract and relax. Generally, the veins, especially in the lower extremity, have valves which prevents the backflow from going backwards. So the blood is kind of pushed towards the heart, withstanding gravity. So what assists this is the muscle contraction of our muscles. That's why exercise, walking is so important in that respect. So the venous sinuses have very little smooth muscles. So therefore they depend on the skeletal muscle system and the valves that exist in this area to assist in pumping the blood back to the heart. Some problems that can occur in these valves is called varicose veins. I don't know if you've ever seen them, but they look like tortuous, twisted appearance of the veins, and this is where the valves are not collapse and therefore you could see the veins, they become dilated and twisted. This is often a basic treatment would be wearing stockings to assist in that return of venous blood In extreme cases, they actually perform surgery on those veins. So now that we're talking about veins, veins are considered blood reservoirs. So veins, the largest portion of your blood at rest, about 64%, is located in the veins. So veins are blood reservoirs. In comparison to systemic arteries, holds approximately 13% of the blood. The capillaries would hold about 7%. Pulmonary vessels about 9%, and the heart holds about 7%. So let's look at this image here. We could see the elastic arteries, which are the largest arteries, and how they compare to the other vessels. Very large tunica media, tunica externa, and they're overall pretty large. And then these are extend into muscular arteries. So these are the larger arteries that include the aorta, the vessels that come off of the arch of the aorta, including the brachiocephalic artery, the carotids, and the subclavian arteries. And then these empty into the muscular arteries, or the distributing arteries. The distributing arteries are carrying this blood throughout our muscles, and then eventually go into these microscopic arterioles. And you can see how the lumen and there's changes in the size. These dump into the capillaries, and this is where all the gas exchange occurs. And then the blood on the way back to the heart goes through the venules, the mid-sized veins, and then the larger veins. So this is an overview of what we discussed today. Make sure you know the names of the different types of arteries and somewhat of the function of what they do. So elastic arteries are considered resistance. Muscular arteries distribute the blood. The arterioles are controlling the rate of blood flow into the capillaries. In the capillaries is where all the exchange occurs. The precapillary venules and the muscular venules is bringing the blood back to the heart. So this is a nice review of everything we've covered. So next we're going to go over the fetal circulation. So let's look at this. Excuse me. So the lungs and the GI organs in the fetus does not begin to function until birth. So the exchange of materials is via the placenta. So oxygenated blood from the mother enters via the placenta via the umbilical vein. So the umbilical vein is carrying oxygenated blood to the fetus. So at this point, some of this blood that enters is going to go to the liver for processing, and some of it is going to bypass going into the liver and going into the ductus venosus. The ductus venosus. So the ductus venosus is going to carry some of the blood into the inferior vena cava, into the right atrium, and some of that blood is going to go through the liver for processing, detoxification, and also going into the right atrium. So the right atrium, in the right atrium, most of the blood is going to go via the foramina valley directly into the left atrium, because the lungs are not functioning yet at this point. Some of this blood can go into the left ventricle and out the aorta. It's going to bypass the lungs, and it goes a little bit will go into the pulmonary trunk. So from the pulmonary trunk, most of the blood via the ductus arteriosum is going to bypass the lungs, and it's going to go into the left atrium. And then from the left atrium, it will go into the aorta, throughout the entire body, and then will return via the blood from the fetus to the placenta via the umbilical arteries. Okay, so that's an overview of the fetal circulation. So let's look at some of the things that we should look at. So first of all, if we're looking at the entryway of this blood via the umbilical vein, one of the bypasses is the ductus venosum, becomes the ligamentum venosum after birth. And then as the blood enters the inferior vena cava into the right atrium, the foramen ovale in fetal circulation will close up to become the fossa ovalis. And then as this blood is going through the pulmonary trunk, there's a connection between the pulmonary trunk and the arch of the aorta, and that's called the ductus arteriosum. That's going to become the ligamentum arteriosum. Okay, make sure you pay attention to all of those. Have a great day.